In this video, we're going to see few important multiple choice questions from molecular biology, B complex vitamins, minerals, detoxification, nucleic acid metabolism. So here we have total of 20 MCQs, which are very, very important from all these chapters. So the first MCQ will be the vitamin required for the carboxylation reaction is. So here we have four options. The first one is niacin, and the second B is pyridoxine, C is biotin, and D is thymine. The correct option is B. So you can see here B is a correct answer, biotin. So here the answer is C. See, the niacin is required for niacin is a vitamin. It gives us niacin coenzymes which are NAD plus and NADP plus. Pyridoxine, the coenzyme form of pyridoxine is PLP, which is required for transaminases. This acts as a coenzyme of transaminases. And biotin here, biotin is a vitamin which is required for carboxylation reaction. How to remember that biotin is only required for carboxylation reactions, I will tell you. So biotin, the letter. So this biotin letter, the vitamin biotin letter starts with B, right? So BIOTIN. So B is a second alphabet. What after B? So third alphabet is C. After B, C. So biotin requires for C. C means carboxylation. So biotin is particularly required for carboxylation. Remember, biotin is required for carboxylation reaction. Let me erase it. The correct answer is C for the first MCQ C. And the second one is secondary hyperuricemia is due to. So the option A, abnormal PRPP synthetase, and B, glucose 6-phosphate deficiency, glutathione reductase variant, and rapidly growing malignant tumors. So the first three, A, B, C, all these are primary hyperuricemia, like they come under primary hyperuricemia. And the fourth one, D option is correct, rapidly growing malignant tumors. This comes under secondary hyperuricemia. So move on to the third MCQ. You can see here the correct answer is D over here, and here the first one is biotin. Now it is clearly visible. So now see the third MCQ. Northern blot technique is used to identification of option A, sequences of DNA, B, sequences of RNA, C, sequences of protein, and D, specific DNA protein interaction. The correct answer will be B. The correct answer is B. Northern blotting is specific is essential for identification of specific sequences of RNA, not DNA. DNA is by southern blotting, and protein identification, protein sequence is by western blotting. So D is just randomly given. So here DNA, RNA, and protein. So RNA sequences will be done by the technique northern blotting. So the correct answer is B. So now move on to the fourth one. Let me scroll it up. Okay. So yeah, the fourth one. Let me erase these things. Yeah. Blastoma. Retinoblastoma can result from a mutation in. The correct option is option D, RB1 gene. About these remaining three genes, I will explain in molecular biology. So the correct answer is option D, RB1 gene. And the fifth MCQ, anti-cancer agent methotrexate inhibits the enzyme. Option A, thioredoxin reductase, B, dihydrofolate reductase, ribonucleotide reductase, and D, thymidylate synthase. The correct answer is dihydrofolate reductase, because dihydrofolate reductase is an enzyme which converts the folic acid, dietary folic acid, into tetrahydrofolate in the liver. So methotrexate is a drug which is advised for cancer patients. So this methotrexate inhibits the formation of tetrahydrofolate from folic acid, so that tetrahydrofolate will not be available for one carbon metabolism. DNA synthesis will be stopped, and cancer tissue will not be developed further. Cell division will be halted. So this is how. So all these three are randomly given enzymes. Okay. The sixth MCQ is which of the following enzyme uses selenium as cofactor? So here you have four options A, B, C, D: hexokinase, transaminase, ferrochelatase, and 5' deiodinase. So hexokinase cofactor is magnesium, transaminase the coenzyme is PLP, and ferrochelatase, no, selenium is not a cofactor of ferrochelatase, and D, 5' deiodinase has a cofactor which is selenium. So remember, selenium is a cofactor for 5' deiodinase. And now the sixth one, let me erase it. Exposure of DNA to ultraviolet radiation can lead to formation of. The correct option is the first option: A, adenine dimers; B, guanine dimers; and C, thymine dimers; and the fourth one, D, is uracil dimers. The correct one is thymine dimers. So we'll explain in later about all these dimers, how they are formed by ultraviolet UV radiation. We'll explain later in a special class. So the eighth MCQ is the vitamin required as coenzyme for the action of transaminases. Remember, the first option is A, niacin; second option B, pantothenic acid; and C, pyridoxal phosphate riboflavin. So niacin, we already discussed the coenzymes of niacin or NAD plus and NADP plus. So it's not. These coenzymes are required for carbohydrate metabolic enzymes and a few other enzymes also. Niacin coenzymes are required as a coenzymes. Pantothenic acid coenzyme is CoA, so which is required, which is central metabolic cofactor. Like pantothenic acid act as a central compound for all the metabolisms. So acetyl CoA, succinyl CoA, for all that, pantothenic acid gives a coenzyme in the form of CoA. So here this C, option C, is pyridoxal phosphate. So this is a correct answer. So all the transaminases require pyridoxal phosphate, PLP. So in the form of PLP, this coenzyme is required for transaminases enzyme. So this is a correct answer. C is a correct answer. And riboflavin gives us FAD plus and FMN coenzyme. This is incorrect, and these two incorrect. So now move on to the ninth one. Which of the following compound is detoxified by the glucuronidation reaction? No, aspirin is not. Methanol is also not. Phenyl acetate is also not. All these, like aspirin, methanol, all these comes under first order reaction, or you can say first phase reaction of detoxification. And phenyl acetate is also phase 2. But bilirubin, particularly bilirubin, these are incorrect. These three are incorrect. The last option, D, bilirubin, is correct. So bilirubin undergoes glucuronidation reaction by glucuronic acid in detoxification. So now the 10th one is, the 10th MCQ is BMR is increased in fever. Yeah, the correct answer is fever, not in Addison's disease, starvation, and hypothyroidism. In all these, it will be decreased. So the 11th one will be, the 11th MCQ is, which of the following mineral deficiency causes Keshan? Sorry for here the mistake. Keshan's disease. The question here, it's not mentioned Keshan, but remember, which of the following mineral deficiency causes Keshan's disease? So here you can see copper, selenium, zinc, manganese are the options. The correct answer is selenium. Copper deficiency leads to Wilson's disease, and zinc, manganese, they have a particular disorders like enteropathica. So all these like zinc related, and manganese also, they have a particular other disorders. But selenium, particularly selenium, has a deficiency which causes Keshan's disease. Okay, and then now the 12th MCQ will be, which of the following enzyme makes a break in stand of DNA to release the supercoiling in a DNA replication? So it's a very easy, super easy, topoisomerase. Topoisomerase is the correct answer. So which prevents from supercoiling of the DNA in replication. So the 13th one is steroid hormone synthesis. Steroid hormones are synthesized from cholesterol, tryptophan, tyrosine, arachidonic acid. So arachidonic acid is a precursor for what? Tyrosine and tryptophan. They synthesize a specialized compounds in amino acid metabolism, some neurotransmitters and all. And the D option, arachidonic acid. From arachidonic acid, we get prostaglandins, and cholesterol is a Correct answer, cholesterol is a precursor for all the steroid hormones. So cholesterol is a correct answer and remaining three are incorrect answers. Okay, cholesterol is a precursor for all the steroid hormones. Steroid hormones are what? The sex hormones and all, adrenal hormones, sex hormones and all. Okay, move on to the further next 14th MCQ. Gene amplification can be achieved by. So gene amplification in a lab level will be amplified by the polymerase chain reaction. Southern blotting technique is for identifying DNA segments. It's incorrect. And DNA fingerprint is also a separate technique. It's incorrect. Gene cloning is also other different technique which is not come under gene amplification technique. So gene amplification technique can be achieved by polymerase chain reaction, PCR. So C option is the correct for 14th. So the 15th one, which of the following is a second messenger? See here, all these are not like CCMP, CTMP, CUMP. The CAMP, cyclic AMP is a correct. The option B is correct. So this is a second messenger in hormone signaling. Remember the B, CAMP is a second messenger. Wherever you see second messenger, the correct answer is CAMP. And the 16th one is the most common form of DNA is the bDNA. The most commonly present DNA is bDNA. The option D is correct for 16th. And the 17th MCQ will be the vitamin that acts as a carrier of one carbon metabolism. So as we all know, very easy one, like the vitamins. From vitamins, niacin, it gives us coenzymes. It will not be involved in one carbon metabolism. And thymine, TPP, of course, it is not involved. It will not be involved in one carbon metabolism. Similarly, pyridoxine, which gives us PLP, it's incorrect. Thymine incorrect. Niacin also incorrect. And folic acid in the form of tetrahydrofolate active form will be involved in one carbon metabolism. So the option A is correct in 17th MCQ. So move on to the 18th MCQ. MCQ is transcription is terminated by. So the correct answer is B. The correct answer is B. Rho factor, RHO factor binds to mRNA. And here the ribosome reaches poly A tail on mRNA will be seen in translation, not in transcription. And the codon on mRNA is AUG. No, incorrect. And codon on the mRNA is UGA, also incorrect. Whether it is stop codon or start codon, rho factor binds to mRNA, then only the termination occurs. So the 19th MCQ will be the cell cycle is regulated by. So cell cycle will be regulated by the cyclin dependent kinase, not by the DNA dependent kinase and availability of nucleotides present of inducers in cell. So A, B are incorrect. The option C is correct and D also incorrect. The 20th last MCQ, which enzyme is used for the preparing a recombinant DNA? So very easy, 11th, 12th standard question. So obviously restriction endonuclease. RNA polymerase is required for synthesis of RNA in transcription. DNA polymerase is required for DNA replication. Topoisomerase is also required for DNA replication to prevent the supercoiling of the DNA. All these are incorrect for this question, and the 20th correct answer is A, restriction endonuclease. Thank you for watching. Please do subscribe if you like it.